Hello, can somebody confirm you can hear me?
I can hear you.
Thank you. Okay, before we begin, are there any questions? All right, today is October 14th, and we should be doing Lab 5, Streaking for Isolation and Culture Media. I also need to tell you that you should read the unknown project instructions file, which is on Canvas, for next week. We'll begin the unknown project next Tuesday. And so you need to know about the project, so I'm giving you the instructions. Let me show you where that is if I can get to it. Come to the main web page. Are you seeing my screen?
Yes.
Great, thank you. Scroll down to week 5, I think it is. Week 5 lab, unknown report, the unknown instructions. This is the file you should have read by next Tuesday. Any questions about any of that? We'll talk about the project on Tuesday. That I think is it for the syllabus, so I can probably shut that down. Don't think there's anything I need to talk about. So let's go straight to the lab manual. Lab 5, cultivation of bacteria, culture media, inoculation, and isolation. There is some reading to every lab. Not too many pages today. You read about culture media, page 157 to 162. A page about obtaining pure cultures. There are some video clips in today's lab. There is no longer a virtual lab because the one we had used Flash, and Flash is not supported and doesn't work anymore. The learning objectives for this lab is 1, understand the importance of proper specimen collection and transport to the lab. 2, explain the 5 principal steps used by microbiologists to process specimens. 3, differentiate between various types of culture media. 4, explain the purpose for streak plate culture technique, aka streaking for colony isolation. And I usually call it streaking for colony isolation. And 5, define the following terms: what's selective media, differential media, and reducing media. Does anyone have a question? All right. The ability to isolate pathogens from a clinical specimen submitted for culture is only as good as the collection technique and the timely transport of the specimen to the lab. Some microbes are sensitive to desiccation, drying out, and some are sensitive to exposure to oxygen, almost all anaerobes, and therefore require a special transport medium. In reality, you're going to need a special transport medium regardless of microorganism you submit, because all microbes will require a transport medium to stay alive. But some have to remove the oxygen, and others make sure the cell doesn't dry out. Once a specimen is received in the lab for a microbiological culture, microbiologists employ the 5 principal I steps to process the specimen. 1, inoculation, 2, isolation, 3, incubation, 4, inspection, 5, identification. So 1, inoculation, that's placing the specimen or the cells on an artificial culture medium to initiate growth. 2, you then have to isolate the growth, so you spatially separate microbial growth into distinct colonies. In fact, you'll need to probably go further and make sure you have a pure species before you study it further. 3, you need to incubate the specimen, and that's simply placing the culture media in an environment having conditions to facilitate growth. We normally incubate our cells in an incubator. 4, and then after growth in the incubator, you then inspect the culture, examining it for presence of microbial growth. You can do some preliminary work on trying to identify it, but you really won't identify it until you perform specific tests to ID a pathogenic microbe that's been isolated. And that's the 5th step, identification. Almost all labs use biochemical tests to identify the species, but there are some labs that will identify the species just by sequencing the DNA. All right, we'll describe the 5 steps in detail after discussing the different types of culture media. You're already familiar with general purpose media, such as Na, nutrient agar, Na, or nutrient agar with yeast extract, and TSA, or tryptic soy agar. These are all media to grow many organisms, but we call them general purpose because they are not selective. They grow many organisms, but they do not grow fastidious organisms. A fastidious organism is one that requires a nutrient to be added to the media before they will grow. And it could be several nutrients. Like Streptococcus pyogenes is fastidious. It will not grow on Na or Na or TSA unless you add 2 ingredients to the media. And usually how we grow Streptococcus pyogenes and many other fastidious microbes is we just grow it on an enriched general purpose media, such as blood agar. Blood agar actually is TSA, but it has 5% sheep's red blood cells added to the media. The sheep's red blood cells provide the nutrients, at least for Streptococcus pyogenes, to grow. Any question about any of that? All right, so an enriched general purpose media is one that has additives added to it to grow fastidious microbes. We also have selective media, and this is a media that has chemicals which inhibit or halt the growth of some organisms, but allow the growth of other organisms. Keep in mind 2 things when using selective media. First, a selective media isn't necessarily perfectly or 100% selective. If given enough time, some organisms that are inhibited may grow and reproduce on the selective media. And second, those organisms which are supposed to grow on the selective media may form smaller colonies or may grow slower on the selective media than they grow on a general purpose media. There are many kinds of selective media, and I actually got this out to show you. In microbiology, we have in our microbiology lab, we have a book that's about this thick. It's actually a little wider than this, and it's actually a little bit longer than this, so a little bit bigger than this, but about this thick is correct. We have a book this thick And in this book, on each page, we have one media. And that's a lot of pages of media. And that's how many different media we have in microbiology that a microbiologist may use. So we have many different media in microbiology. Let's talk about a selective media. Mannitol salt agar is a selective media. McConkey's agar is a selective media. We have another type of media that we call a differential media. These media contain chemicals that produce visible differences among different groups of organisms when they're grown on that medium. These differences are unrelated to how well the organism can grow on the medium. Usually, the dissimilar bacteria can visually be distinguished from each other. Like they'll have a different color, or they will do something different in the media, like they will have a different halo around them. It's usually a visually distinguished difference. Lastly, we have a reducing media, and this is media to promote the growth of anaerobic bacteria because these bacteria require low or no oxygen conditions in order to grow. It's important to note that a culture media can be classified into more than one category. For example, McConkey's agar is both selective and differential. And blood agar is both an enriched general purpose media and a differential media. Any questions about any of that? All right, here we're looking at McConkey's media. It is selective so that gram negatives tend to grow on this media and gram positives tend to not grow. You don't need to know why, but the gram positives tend to not grow because the media has crystal violet in the media, which gets stuck in the cell wall of a gram-positive cell and then doesn't allow the cell to grow. It also inhibits gram-positive cells because it has bile salts in it. Bile salts you may be familiar with. It's in your intestines. We produce it to help digest certain foods. And gram negatives tend to grow in bile salts because they grow in your intestines. Gram positives don't like the bile salts, and it inhibits most of their growth. So that's how McConkey's is selective. It's differential on the basis of lactose fermentation. The sugar lactose is in this media, and if the bacteria can ferment the sugar lactose, it will produce acidic byproducts, which will change the pH of the media, giving the pink color. And you can see the pink color both on the colonies or on the growth right here, as well as around the growth in the plate, the pink of the agar right there. And that's just from the decrease in the pH from the fermentation of the sugar lactose. Any question about that? On the other hand, if the cell is a gram negative, it will grow on McConkey's agar, as this one did. It will not be pink. The bacteria will be its normal color, which for this one is an off-white color. But whatever the bacteria's normal color will be, it will be that color. Like if you were to grow I'm trying to think of one that doesn't, Pseudomonas on this McConkey's agar, it would be, well, a bluish green in color, at least Pseudomonas aeruginosa. It may only be in the colonies, maybe not around it, but it would be in the colonies. Micrococcus luteus will not grow on McConkey's agar, but let's say it would. Micrococcus luteus is yellow in color, and there are other species which I don't remember what their name is that are yellow in color. So I'm just saying Micrococcus luteus. If it were to grow, it would be yellow in color because that's its normal color. Of course, Micrococcus luteus is gram-positive, so it should not grow on McConkey's agar. And then here's blood agar. It is both a enriched complex media, enriched general purpose media, meaning an enriched media, because it has blood in it to supply nutrients that the cells may require, like Streptococcus pyogenes, can grow on blood agar, but cannot grow on a general purpose media. It's because the blood provides the nutrients the Streptococcus pyogenes requires. So that's what it is, enriched media. It's also a differential media, and it differentiates cells based upon their hemolytic activity. You can have cells which have gamma hemolysis, that's no hemolysis, shown here, and that's actually the lower letter for gamma in the Greek alphabet, like that. And you can see the blood being red all the way up to the bacteria. We also have alpha hemolysis, and that is partial degradation of the red blood cells, creating the darkening or sometimes a greenish tint around the bacteria cells. In the alpha, we have the cells growing, so the bacteria are off-white in color. And you see initially the red, and then you have this dark ring around the cells, and that's alpha hemolysis, which is also inside the alpha, the darkening. Sometimes when you look at alpha hemolysis, it could be a greenish color. Here it's a darkish color. And then we have, whoops, didn't mean to move that. Let's move that back. There we go. We also have beta hemolysis, that is complete hemolysis. So in the beta here, the Greek letter beta, the white color is the bacteria growing, and these bacteria have beta hemolysis or complete hemolysis, creating the clear zone around the bacteria and inside the bacteria. You can actually see through the clear region. You can hold this up to the light and see the light bulb through the clear region. I sometimes hold it up to the clock and then try to tell the time because you can see through the clear region. Any questions about the three different hemolytic activities or why blood agar is differential? All right, a note of the beta hemolysis. Around the beta hemolysis, meaning the area where they have complete hemolytic activity, you'll notice a ring of alpha hemolysis, the darkening. And that's normal. You normally have alpha hemolysis outside of the beta hemolysis. So the point is blood agar and McConkey's are both, well, McConkey's is both differential and selective, and blood agar is both an enriched media and a differential media. The table here shows you the different types of media and then some of the media, the ones we'll talk about in this lab. So for the general purpose media, it grows all bacteria, non-selective. As long as it's not fastidious, it will tend to grow on a general purpose media, including TSA or tryptic soy agar, Na or nutrient agar, or Na, and that's nutrient agar with yeast extract. We also have a differential and enriched medium, and that's blood agar or BAP, showing you allowing fastidious growth of the bacteria, meaning a fastidious species, at least some of them, like Streptococcus pyogenes. And then you can also determine the hemolytic activity of the bacteria on blood agar. So that's why it's differential. And then we have CAP or chocolate agar. It is a enriched general purpose media. promoting the growth of bacteria that would not grow on a general purpose media. CAP is not differential, by the way. And then we have selective and differential media. We have three of them: McConkey's or MAC, eosine methylene blue or EMB media, and hectone agar or HE agar. They are selective because all of them select for gram negatives and select against gram positives. So the gram negatives should grow on these three media. The gram positive should not. And they're all three of them differential on the basis of lactose fermentation. If they can ferment the sugar lactose, they will be a different color than if they cannot ferment the sugar lactose. If they cannot ferment the sugar lactose, the bacteria will be their normal color, whatever color they should be. And many bacteria are off-white or white, meaning they lack a color, no pigmentation. If they can ferment the sugar lactose on McConkey's agar, the bacteria culture itself, meaning the colonies, will be pinkish, and then the growth, the agar around the colony will also be pinkish. If the bacteria can ferment the sugar lactose and it's growing on eosine methylene blue, the bacteria will either be a dark color like a black, real close to black, or it will be metallic green if it can ferment the sugar lactose on EMB. And if the bacteria can ferment the sugar lactose and it's growing on HE plates or hectone agar, it will be a salmon color. For those of you who do not know, salmon is a yellow-orange color. All right, our next media is PEA or phenyl ethyl alcohol. This media is selective and it selects for gram-positive bacteria, selecting against gram-negatives. So if you have a gram-positive, you want to grow it on PEA. If you have a gram-negative, you want to grow it on one of these three: MAC, EMB, or HE. Our last media is mannitol salt agar or MSA. It is both selective and differential. It is selective because it has 7% sodium chloride in the media, and only bacteria that can tolerate or enjoy growing in 7% sodium chloride can grow on MSA. Most bacteria cannot grow if the salt, that's MSA, the 7% sodium chloride, if the salt is above 2%, most bacteria cannot grow. And MSA has the sodium chloride at 7%. So Micrococcus luteus can grow on MSA. Staphylococcus aureus can grow on MSA. And there is one gram-negative. I think it's Citrobacter aerogenes? No. Citrobacter cleoca, cleoca, cleoca. If it can grow on MSA, it can tolerate the 7% salt. So that's selective. It's also differential on the basis of mannitol fermentation. If the bacteria can ferment the mannitol, they will have a yellow halo around the colony. The colony itself may turn yellow. But like Staphylococcus aureus and Micrococcus luteus are already yellow, so you cannot tell by the colony color, but you can tell by the halo around the colony. It'll be a yellow halo. If it does not ferment the sugar mannitol, the halo around the bacteria will be the same color as the background of the plate, meaning the same color as the media in the background. Any questions about any of these media?
I have a comment. So document that you have on Canvas is slightly different than this table that you have here.
What's the difference?
It doesn't really mention much about the differential media in the table on Canvas, like in that document that you have for this module.
Let me take a look at that. That might be wrong. Are we on lab 5? Yes. I should probably download it. It'd be easier to see, but I've got it here now. Mannitol is selective and differential. McConkey's is selective and differential. Do I talk about more in mine?
There was just some comments in the TSA with 5% sheep blood one. And that one, yeah, that one.
So this one here has something different, is what you're telling me.
Yeah, it says that differential on the basis, and that one didn't have any comment about that.
All right, I'll try and upload this file here. Oh, it's really just talking about, oh, how it's differential. Yeah, I'll try and upload that. Let me see if I've got that. Um, because I'd rather not upload the one I'm talking from because I have it highlighted, the points I want to cover. But let's see if I have it. Oh, there we go. No, I got to change it. So let me make a note of that. And then I'll try and get that done after the lab. All right, let's leave that up and it might help me remember to do that. Wrong one. All right, thanks for pointing that out. I made a change here and apparently didn't um, upload it. I don't know why this thing jumped around like that. Okay, let's talk about inoculation. That's just putting the cells from like a clinical specimen on the media and then getting them to grow. You then do isolation. And Louis Pasteur first developed the concept of isolating a pure culture. How he did it was he took the original sample and then made various dilutions of it, like a 1 to 10, 1 to 100, 1 to 1,000. And then he made 10 tubes of each dilution. And then he inoculated each tube, so he might have 100 tubes here. And if he ever got one tube out of 10 having growth in it, and 9 tubes of this dilution having no growth in it, he correctly surmised that the tube with one tube having growth was likely to be formed by one or a few cells and was likely to be a pure culture. And he was correct, so that he was able to isolate pure cultures and able to study them. It was a very difficult way of getting a pure culture. You'd have to have a original sample, a mixed culture, and then make dilutions of it, many different dilutions, and then you have to replicate plate 10 tubes for each dilution. And then you're looking for the one dilution where there's one tube that has growth in it and 9 tubes having no growth in it. Robert Koch developed a much easier way of getting a pure culture. And what he did was he streaked cells on a solid medium. And if you streak the cells through a solid medium to get isolated colonies, and you take cells that went through 2 colony isolation events, you have a very high chance of getting a pure culture. It's greater than 95%. In microbiology, we never get 100%, but it's greater than 95% if you take cells that have gone through 2 colony isolation events. So what you want to do when you're streaking out is in one of the quadrants, when you're streaking, you're putting down individual cells. They'll be isolated from each other and then will grow up and form isolated colonies away from each other. So that's the purpose of streaking, to get isolated colonies. Let me scroll down just a little. If you take an isolated colony here, it's only about 70% chance that it will be a pure culture. 30% of these colonies will be a mixed culture from 2 or more cells. And that's because when you put cells down on the plate, when you're streaking, sometimes, most of the time, you'll put down 1 cell, but sometimes you'll put down 2 cells or 3 cells or a clump of cells. Wherever you have especially a clump of cells, you're likely to have a mixed culture in this colony. It may look pure, but you have to streak it out on another plate to make sure it's a pure culture. And that's what Dr. Koch did. He streaked it out one time, took an isolated colony from this plate, and then took the cells from that isolated colony, streaked it out on another plate to get isolated colonies. And then if you take from an isolated colony on the second plate to set up your culture, the culture is likely to be a pure culture with a greater than 95% chance of it being a pure culture. Any question about any of that? Why did I lose the position again? Whenever I touch that, it tends to jump. All right, let's talk about streak plating. To do this correctly, what you do is you sterilize your loop by putting it either into the flame or the bacto incinerator, and then putting it into your bacteria. So let's pretend this is the bacteria and it's a mixed culture. You cool the loop on the inside. I'm not going to touch the inside because this is a dirty pencil. This is my glass. Let me get something that I don't care if I contaminate it. There we go. So you cool the loop by touching it on the inside of the tube, and it should be sterile because the tube has been sterilized and it's covered with a cap. So you touch it on the inside of the tube, and then you pick up the bacteria on your loop, and then you put it on the plate in quadrant 1, and you streak it on quadrant 1. I'll use the mouse. Streak it in this type of direction. You should be streaking about 20 to 30 times in each quadrant. So this is only showing you like maybe 6 or 8 times. You'll be streaking 20 to 30 times. And it's too hard to draw that many streaks out. Although here you got it there. The first streak will have the highest concentration of cells. The last streak in quadrant 1 will be the least concentration of cells. And that's because as you're streaking, more and more cells are coming off the loop. So this will be a lower concentration of cells. You then take your loop, pass it in the flame or in the back-to incinerator, and then you cool the loop by touching it in quadrant 2. It's best to touch it in a place you're not going to streak, but someplace in quadrant 2 to cool the loop. And then you take your streak and you streak it through into quadrant 1 3 or 4 times. What you should do is streak it in quadrant 1 in the dilute regions of quadrant 1. So you don't want to streak it up here because that's the most concentrated region of quadrant 1. What you want to do is only streak it in this region here. Can you guys see my mouse? Anyone?
Yes.
All right. So you streak it in the low regions. You don't streak it in the high regions. And then you streak it out and you form quadrant 2. Once again, you streak it about 20 to 30 times. Then you take the loop and you put it in the back-to incinerator or the flame to sterilize it, and you cool it by touching the auger in quadrant 3. And this is actually shown a little better than quadrant 2, and that is you begin the streak in quadrant 3 going into quadrant 2. But you should only go where it's dilute in quadrant 2, meaning in this region here and this region here. You should not be coming up to the region in quadrant 2 where the bacteria are heaviest, and that would be this region here. So you should not be coming up this far. You should only be in this area. And you streak it in quadrant 2, 3 or 4 times. And then you streak it out on quadrant 4. Quadrant 3, sorry, quadrant 3. And then you sterilize the loop once again in the flame of the back-to incinerator. You cool it by touching the bacteria in, not the bacteria, the media in quadrant 4, especially in a region you're not going to streak it on, like maybe right here. And then you begin your streak in quadrant 4, going back to quadrant 3. And this one is actually shown well, and that is they're only streaking in quadrant 3 in the dilute regions of quadrant 3, going into quadrant 3 about 3 or 4 times. You do not want to go in the high region of quadrant 3, just the low regions. And then streak it out about 20 to 30 times, not shown. And you will get isolated colonies, either here as shown in this case, but if the bacteria were to be a little more dilute than we started with here, you could get the isolated cells being put down in quadrant 3, and then you get the isolated colonies in quadrant 3. And if it was even more dilute than that, you could get the isolated colonies on quadrant 2. And if it was very dilute cells to begin with, you could actually get your isolated colonies on quadrant 1. If you were to put down isolated cells in quadrant 1 and obtain your isolated colonies in quadrant 1, what would you likely have in quadrant 3 or quadrant 4? What would you likely have in your streak? Would you have isolated colonies? Would you have a whole bunch of growth in quadrant 4 like here? Would you have
No.
isolated colonies there? What would you have in quadrant 4?
Isolated colonies.
No, you have the isolated colonies in quadrant 1.
So would there be anything in
There wouldn't be
three?
anything likely in
Okay.
quadrant 3 or 4 because when you go to form quadrant 2, you may not pick up anything. And if you do, it will streak out in quadrant 2, giving you a few isolated colonies in quadrant 2. And so quadrant 3 and 4 will probably be nothing. Okay? I'm sorry I filled in the answer for you, but anyways, if you have isolated colonies in quadrant 1, you're not likely to have anything in quadrant 3 or 4 because the cells are so dilute that when you're streaking the next quadrant, you're likely not to pick up anything. All right, the next step is incubation. Recall that culture media contains nutrients for in vitro growth. Microbiologists must provide an appropriate physical environment, such as an incubator for putting our plates in, like this one here. This incubator is a fancy one. It can control how much oxygen the cells are going to be exposed to, how much CO2 the cells will be exposed to, what the temperature will be, what the humidity will be. We have that container in there, and you put water in there, and that'll make the air humid in the incubator. And then the time. The time is just decided by you when you put the cells in and when you pull the cells out. So you can vary the time depending on obviously your schedule and when you wish. The incubators in our lab are simple ones, and they only can vary the time, the humidity, and the temperature. And you set the temperature for the incubator. Usually one incubator is set at one temperature, and then it stays at that temperature until the temperature is changed. Humidity, we get that by adding water to this pan in the incubator, and then the heat of the incubator makes the water humid, and then we have humidity in the incubator. Humidity is important because if you had a non-humid incubator, meaning no water in the incubator, then the plate can only stay in the incubator for about a week before it starts to dry out. If you have a humid incubator, the plate will last at least 2 weeks and maybe 3 weeks in a humid incubator. And once the plate dries out, the cells will start to die. So humidity is important, but obviously temperature is important too. We have most of the incubators in the lab either set at 30 degrees Celsius, 35 degrees Celsius, or 37 degrees Celsius. You then have 4 inspection. This is looking at obvious visible colony morphological differences between different species of bacteria. Microbiologists can describe the appearance of the colonies, their variety, their size, their shape, their margin, their elevation, whether they're pigmented and what that pigmentation is. But you're unlikely to be able to state the species just based upon the colony characteristics. We then have to identify the species by doing further biochemical testing. At least most labs do identify the species by biochemical testing. It is possible for a lab to identify the species by sequencing their DNA. That's just not normally done. You should know the terms of this lab. And then we have some videos for this lab. Everything we do will be done online, but you should understand the procedures and the terms we've discussed in this lab. We have a little video for you to watch on inoculating an agar plate to obtain a streak plate culture technique, meaning you're streaking for colony isolation. You only need to watch this video until 4 minutes and 15 seconds. After that, you do not need to watch the vector. You should note that in the streaking of this video, the author of the video has only streaked out 3 sectors instead of the 4. It doesn't matter how many sectors you should streak. Most people do either 3 or 4. And then we have a little video explaining beta hemolysis, alpha hemolysis, and gamma hemolysis on blood agar plates. We have a little video on McConkey's agar explaining McConkey's agar. In this video, you only need to watch it until 3 minutes and 50 seconds into the video. After that, you can shut the video down. And then we have a little video discussing mannitol salt agar plates? All right, that's the lab. Let's go ahead and go to the worksheet, and I'll go through the questions. So the worksheet to Lab 5, question 1. Describe a natural circumstance where Staphylococcus species of bacteria might benefit from salt tolerating, meaning why would Staphylococcus benefit to tolerate salt? And you cannot state so that it'll grow on an MSA plate because that is not a natural circumstance. Any question about that? That's a laboratory circumstance. All right. Question 2. How might hemolysis benefit a hemolytic organism? If we have a hemolytic bacteria that can lyse the red blood cells, why could that benefit the bacteria? And then 3. Imagine you have a culture containing a single bacterial species, but the culture is unidentified, and it could be one of several possible species. Describe a scenario where you use mannitol salt agar, blood agar, and EMB plates to help identify the unknown bacterial species. You must name 2 properties to each of these plates to help you identify the species. Meaning in this question, you need to answer 6 different properties, 2 from each plate. 4. What is the main goal of streaking for colony isolation? 5. What happens to the growth on the plate if you do everything correctly, but you forget to sterilize the loop in between sectors when streaking for colony isolation? Be specific. Discuss the growth on each sector. I will not know the correct answer if you do not discuss how much growth is on each sector. And once again, it also says you're doing everything correctly. So you normally sterilize the loop before you streak out sector 1. So that you do. However, you're not sterilizing the loop in between sectors 1 and 2, 2 and 3, and if you have a 4, sectors 3 and 4. Any question about question 5? All right, question 6. What happens to the growth on the plate if you do everything correctly, but you transfer from stock culture to inoculate each of the sectors? So you are sterilizing the loop before you do sector 1, and then you streak out sector 1, and then you sterilize the loop because everything is done correctly, and you go back into the stock culture to streak out sector 2, and then you sterilize your loop, and then you go back into the stock culture to streak out sector 3. So what's going to happen? You need to be specific, discussing the growth on each sector. If you don't discuss the growth on each sector, it'll be at least partly wrong, and it may be completely wrong. 7. Suppose you successfully streak a plate and obtain isolated colonies of 2 different bacterial species. That is, you streaked a plate from a mixed culture. How can you use this plate to create a new pure culture of only one of the bacteria species? Be specific in your answer, meaning you need to talk about everything you need to do to set up a pure culture from one of these 2 species. Any questions about that one? 8. In the table below are a variety of agar plate media. Denote which each medium is selective or non-selective, selective or differential media by putting an X in the appropriate box. I will warn you that some of them have 2 X's for the media. And I'll give you a hint that none of them are both non-selective and selective. They're either non-selective or selective. So the most you'll get is 2 excess on each media. So tell me, nutrient auger, put an X here if you think it's non-selective, put an X there if you think it's selective, put an X there if you think it's differential. Any questions about any of this? And the table in the lab module gives you the answers for these, by the way. You may have to read, but you can find the answer eventually. And then 9, which auger plate or plates were inoculated without using a streak plate technique? Plate A, plate B, or plate C? Any questions? All right, it's now 7:23. I will be here until 8 o'clock or the last student logs out to help you with any questions. Go ahead and get started on the lab.